Today, we will be talking about yeast-3 hybrid. Yeast-3 hybrid is an experimental method used in biology to test for protein to RNA interactions. This can be used to test for known proteins' functions or molecular targets thereof. One major use for yeast-3 hybrid is in the pharmaceutical industry. Yeast-3 hybrid can be used in the pharmaceutical industry to test for potential drug targets. This is very important in experimenting with new drugs to gain a full scope of how they may help or hurt a person. In order to physically see whether the yeast-3 hybrid is interacting, one must use reporter probes. This is a gene that, when expressed, will lead to the production of a fluorescent-colored protein, allowing scientists to confirm if there is a protein to RNA interaction. If there is no fluorescence, then there was no interaction, and if there was fluorescence, then the interaction was present. The way that yeast-3 hybrid works is relatively simple. By introducing a bait and a prey, which are your RNA and proteins you are testing for, you introduce them into yeast colonies. Once this is done, the RNA of interest will be bound to a DNA binding domain, or DBD, that is in the promoter region of your reporter gene, and your protein of interest will be bound to a transcriptional activation domain, or AD, bound to the reporter gene itself. If the bait and the prey are able to interact, this will complete an RNA bridge, thereby activating the reporter gene, leading to a fluorescent protein or blue yeast colonies being formed. If this gene remains inactive due to the RNA protein not interacting, the yeast colonies on the media will be white. This is how you confirm whether or not your RNA and your protein can interact. Yeast-3 hybrid is a useful technique, but as it is still a relatively new technique developed in recent years, it does still have some limitations. Thank you for watching.